Okay, so this is an introduction to kind of basic anatomy terminology, which we'll also go through more in lecture. We kind of did the review of systems today. Um, if we talk about anatomical position, it's important to orient ourselves with the body and always make sure we're talking about a person in anatomical position. And it's defined as the body standing straight with both feet flat on the floor, with palms forward facing, fingers extended, and the thumbs facing to the outside. So in this position, the thumb is pointed away from the body and the pinky is closest to the body. The head is extended and forward facing. It's important to talk about anatomical position because we're going to be describing structures and where they're located in relationship with each other. And as you guys go into a healthcare related field, you'll always want to examine talk about a person and write your notes about your patient based on what's wrong with a particular structure in their body. And you'll always reference anatomical position to show you where that structure is located in reference to something else. So you're supposed to demonstrate anatomical position if you want. I won't make you do that here. So some terms we use, some anatomical terminology we use to describe structures on the body in anatomical position are listed here. And I believe these figures are all taken from the lab manual. So if you want to download that PDF copy of the lab manual, these figures are taken directly from there. So the word superior or cephalic has to do with anything pointed more towards the top. Inferior and caudal would be a structure further below. So an example of how I would use these two terms is I could say the ankle is inferior to the knee. Or I would say the tarsal region, which are your ankle bones, the tarsal region is inferior to your patellar region. So that's a way I would use the word inferior to describe the location of two structures on a body. And again, this is important because you'll see these words in notes. Now they're all computerized, but as you're working with a patient, you'll see the words proximal, distal, lateral, medial, dorsal, ventral, all used to describe locations of something on a patient. The midline of the body is always right down the middle of a person when the person is facing forward at you. And we call a structure medial if it's located closer to the midline, and lateral if it's located away from the midline. So I would say the umbilicus, your belly button, is medial to your forearm because the belly button is more medially located to the midline than your forearm is. Another way to use medial and lateral, those terms, is I would say this person's thumb is lateral to the pinky because the thumb is further away from the midline than the pinky is. So anytime you use the word lateral, you're describing something further from the midline, and medial describes something closer to the midline. Proximal and distal are two other terms we use. Distal is any structure further away from the point of attachment, and that's in reference to the trunk of the body. So usually your fingertips, your toes, those are all distal features. And proximal is anything, think proximity-wise, closer to the point of attachment or closer to the trunk. So the shoulders are proximal to the wrist. The wrist is distal to the shoulders. Do those make sense? The toes are distal to the knee. The knee is proximal to the toes. You could use inferior to describe those same things as well. What else was I going to say? Oh, rights and lefts on the body. So the right and left side of the body is always described as how it is right and left on the patient themselves. So the right side of this person that you're viewing on the left side of your screen is not your right, it's the right of his body. And this is important because if you're a surgeon or scrubbing in on a surgery and he's about to do a surgery on the right forearm, you guys hear stories of this. They do surgery on the wrong limb. So the right side of the body is always the patient's right side, and the left side of the body is always the patient's left side. So this is his left arm, the left leg, the right arm, the right leg. This will be important when we talk about the chambers of the heart, because the heart chambers are called right atrium, left atrium, right ventricle, left ventricle, and it's always rights and lefts depending on how it's in the patient themselves. So it's their right and their left. Students always get that confused, so make sure you keep that straight, right and left. And then ventral just means anterior, closer to the front. Dorsal means posterior. So one example, the buttocks or the gluteus maximus muscle is dorsal to the umbilicus, your belly button. The nose is anterior to the shoulder blade, your scapula. Okay.
Professor?
Yep.
Would you mind doing a quick example again of proximal and distal, please?
Yes.
My apologies.
Yep, no worries. Thanks. I appreciate the question. So proximal and distal are one of the trickier ones. So proximal is describing a structure that's located closer to the point of attachment or closer to the trunk of the body. And the trunk of the body would be anything like the belly button, chest region. So the shoulders are proximal to the elbow because the shoulders are closer to the point of attachment or closer to the trunk than the elbow is. The fingers are distal to the wrist because the fingers are more distally located. They're further away from the point of attachment or further from the trunk.
I see. Okay. Thank you so much.
Yep. No, that's did everyone understand lateral and medial too? Those can be confusing. Another one, the shoulders are medially located to your neck because your shoulders are both further away from the midline, and or laterally located, excuse me, and your neck is medial to your shoulders because your neck is more closer to the midline. Sorry, I'm just seeing the chat here. Oh yeah, yeah, attendance. If you could write your attendance just right here. We're missing like 3 people. I think I know who they are though. Okay. Yeah, so these terminologies you should be familiar with. You should understand them and how to describe structures in relationship to them. This tells you the directional term and the definition if you want to review that there. And then this is surface anatomy describing different body areas, and it shows the anterior and posterior aspects of body surfaces. So the anterior side, again, is the patient's anterior side. So, and their posterior side is on the posterior. This is what that first exercise in the virtual lab takes you through. I think a lot of the matching terms. And again, it's important to learn this and learn it well, especially if you're going into a health-related field. Learn it for this class, but also learn it for life in your future career, because you'll be reading notes on patients talking about the frontal region or the orbital region. The axillary region has to do with their armpit. Once you guys start to get familiar with these words and these terms, you'll start to be able to understand names of structures better. So there are axillary arteries and axillary veins, and where do you think they're located? They're located in the axilla region, which is the armpit region. The carpal region, you have carpal bones, and those are your wrist bones located in the carpal region. The crural region, the tarsal region, the femoral region is you have femoral muscles, but you also have the femoral artery that goes through the femoral region. So even just learning this slide really well will serve you well in learning the basic structures of the body later, whether they're muscles, bones, arteries, nerves, veins. A lot of them are named for what region they're in. You have a frontal bone, which is your forehead region, your forehead bone. Orbital has to do with eyes, your nasal region. Buccal, you have a buccinator muscle. That is the muscle that helps you blow an instrument. The mental region is your chin, oral region. Nuchal is the back of your neck. Occipital region is the back of your head, and you have an occipital bone. So just be familiar with these body surface region names. And this gives you the body regions, their term and their region or where they're located. This looks at the planes of the body, and this is important especially if you're reading an MRI or an X-ray. Well, an X-ray would always kind of be front to back, not necessarily, but all medical professionals are familiar with using these body planes. And it's just basically how you can slice a body. And it's important again for MRI imaging, because MRIs are images. They put you in this really claustrophobic tube and they can take an image of your entire insides of your body. And this is really good for helping diagnose disease. You can find tumors this way in brains, the rest of your body. So how do we cut the body? We have 3 ways we kind of slice the body, which is kind of gruesome, but not really, just if you're looking at an MRI. So the cross section or transverse plane is any cut or plane that divides the body into a superior and an inferior portion. So this cross section or transverse plane, it could be taken up here to go right through the eyes, or it could be taken down here by the ankles. So it's any time we would cut the body into a top half and a lower half, or a superior and inferior portion. So that's a transverse plane. The mid-sagittal plane is one of the easier ones, because mid-sagittal, think midline. So this would be a cut through the body right down the midline of the body. So that's the mid-sagittal plane. And the coronal, or the frontal plane, is a cut through the body that would divide it into a anterior section and a posterior section. So normally I show this in class, but if you go down the side, this would be a cut down right my side to separate my front from my back. So those are the 3 kind of major body planes. And as you're looking at an MRI, they might list, well, this is a transverse section. So you'd be looking at something in this transverse section view. Or a mid-sagittal section. If you think of cutting this man in half, you'd be looking at his 2 halves of the body from a mid-sagittal plane view. So that's why learning these planes are important. There's cavities in the body, so your organs aren't just like hanging out with each other, rubbing up against each other. They're all in enclosed cavities, which keeps everything really safe. The cranial cavity, what do you think is in there?
Your brain.
What about the spinal cavity? I guess it tells you right there. Spinal cord. The thoracic cavity would be your lungs, heart, trachea, esophagus. What's your trachea?
That's your neck bone. Right.
Not your neck bone, but it does go down your neck. So you're close.
Isn't that the vocal area and breathable tube?
Yeah. Yep. So when you breathe air, the air goes down the trachea, and food goes down your esophagus into your stomach. The abdominal pelvic cavity is divided into an abdominal cavity, which would include your liver, your stomach, your intestines, and then your pelvic cavity would include your bladder, your reproductive organs. Between the thoracic and the abdominal pelvic cavities, we have the diaphragm, which is your most important muscle associated with breathing. In the polio epidemic in the early 1900s, polio caused paralysis of the diaphragm, and without it, these kids couldn't breathe. So they were put in like these iron lung chambers. Maybe we'll talk about that more when we get to respiratory system, but that's your diaphragm. So if I ask you a question here, how would you describe the abdominal cavity and the pelvic cavity using those directional terms? Can anyone give me an example?
Abdominal cavity is superior to the pelvic cavity.
Good. Did everyone ever understand that? The abdominal cavity is superior to the pelvic cavity, and the pelvic cavity is inferior to the abdominal cavity. What about describing the thoracic cavity versus the spinal cavity?
Well, this will fall under frontal and rear view, right?
Yeah, so do you remember those directional terms?
It's anterior.
Anterior versus posterior, yep. Or what were other 2?
Ventral.
Ventral and dorsal, yes. So just take a look at this one more time here. Dorsal would be posterior and ventral would be anterior. So we could describe the abdominal cavity as ventral to the spinal cavity. Okay, moving on. Alrighty. And then I think we're here looking at some more body cavities. The mediastinum is just a central body cavity located within the thoracic cavity that includes the heart and the trachea and the esophagus. So the mediastinum is this entire cavity. And within the mediastinum cavity, you have a pericardial cavity, which is just the heart. The pleural cavity includes the lungs. So again, these are just enclosed spaces that keep these organs safe. So this is all the thoracic cavity. The pleural cavity is the lungs. The mediastinum cavity is the trachea and esophagus up here, and it includes the heart. And the pericardial cavity is the heart itself. And this is a look at a model in the lab. Again, these are pictures taken from your lab manual, what's located in each of the cavities. There's also major blood vessels in the mediastinum. And this is an X-ray chest view of the cavities and where everything would be located. Then we get into quadrants of the abdominal pelvic cavity. So we divide the body, especially the abdominal pelvic, into quadrants. And again, this is used for medical purposes. If someone is undergoing surgery or is complaining of pain, they would say this patient is complaining of pain from the right hypochondriac region. Well, everyone will know what that is. It's this region up here. Hypochondriac is up here. Epigastric region. The umbilical region is around the umbilicus. And you can see the right iliac, left iliac, right lumbar, left lumbar. So these are just regions of how we divide the abdominal pelvic cavity. A little bit broader division is just right upper quadrant, left, right lower quadrant, and left lower quadrant. You'll see here already how rights and lefts are important to remember. So remember that the right side of the patient is always the right side of that person's body, not how you are seeing it. And the left side of that person or patient is the left side of that patient's body.
Professor?
Yeah.
For the different regions on the right side of our screen right now,
Yep.
Do those depend on, do they change based on sex and gender? Or are those like pretty solid for as far as the regions?
So yeah, regardless of male and females, the regions are the same. So I'm just gonna mute you guys. I hear myself talking on someone's screen. So yeah, regardless of gender, they are the same because they don't really include the reproductive organs, what are in them. So obviously within these regions, there might be a different anatomical structure, but the regions itself are named the same regardless of gender. Good question. Some people, their large intestine, their colon, most of us, our large intestine starts on the right side, transverses, and goes down on the left. Some people's large intestines are flip-flopped, and that can be tricky if someone complains of pain. Normally an appendix is in your right iliac region. So if you've ever had appendicitis, you'd have pain down here. But if someone has their intestines flip-flopped, that's something medical professionals would look for. Maybe their intestines are flip-flopped and the appendix is down there. Getting ahead of myself. We'll get into that in digestive. Good question though. So these are some quadrants and structures within the quadrants of those, the umbilical, not the umbilical region, but the abdominal region. So you can kind of see what is generally located, and you can see regardless of male and female, what's located in each quadrant. Okay. This is just another view. So this, again, this first lab is to just get you oriented with what are some of the major organs in the body and where they're located. So just some of you might know this already, but you should be able to label a picture like this and know where all the organs are. So your right lung and left lung with the heart in between. Your diaphragm separates out that thoracic cavity from the abdominal cavity below it. Here's your stomach. It hangs out on the left side of the body, right underneath your liver. The gallbladder will be right underneath your liver, and we'll talk about what each of these things do when we get to them. Here's the cecum, which is the start of your large intestine. The appendix would be a little finger-like projection coming off of the cecum. And then here's your ascending colon, transverse colon, descending colon with a small intestine in the middle. How could we describe the heart and lungs using some of our anatomical direction terms?
The heart is medial to the lungs.
Good. And the lungs are lateral to the heart. Very good. Does that make sense to everybody? Hopefully. Okay. All right, so this is an overview of body organ systems. We kind of covered this in lecture, but you should have a general understanding what the 11 systems are, some of the structures that are in those systems, and just a brief understanding of their functions. We'll go over their functions more in detail, but off the top of your head, if I say cardiovascular system, you should know, well, that's your heart and your arteries and your veins. You should know that the cardiovascular system transports substances throughout the body. Muscular system, so be able to understand the systems. What are some that are less common? So your integumentary system is your skin, hair, nails. So your skin, hair, and nails help with body temperature, vitamin D production, excreting waste products through sweating. Skeletal are your bones, cartilage, ligaments. They help to protect and support. Your muscular, you have different types of muscles throughout the body that help with movement, but also pumping blood and helping move things through your digestive tract. Your nervous system recognizes stimuli and adjusts accordingly. So these will be all your brain, spinal cord, neurons. Endocrine is all of your hormones and everything in your brain that controls your hormones. Long-term regulation of homeostasis. We'll talk about more what homeostasis is on Thursday. Lymphatic system are all of your lymph vessels. It returns lymphatic fluid back to circulation. It also plays a role in your immune response. But your lymphatic system, again, I call it kind of the catch-all system because it plays a role in just returning all the excess fluid, cleaning it, and back to circulation. Respiratory system helps to bring in air and exhale carbon dioxide. So you breathe in oxygen because all of your cells need oxygen. And then you exhale carbon dioxide, and CO2 is just a waste product of all your cells. Then your digestive system helps with taking in food, absorbing all the good stuff out of food, and getting rid of anything your body doesn't need. And then the urinary system filters the blood. Your kidneys are just really important organs in the body. So you also want to take care about what you put into your body to not make your kidneys work too hard, because you don't want to go to dialysis centers twice a week when you're older. It's tough. And then the reproductive system, the big thing that helps with hormone production, so testosterone and estrogen during puberty, also sperm and egg production. And females, I guess, developing a baby for birth. Okay, so that's the first exercise. And I'm just gonna go, the second exercise is a little bit shorter. So we're gonna go through exercise 2, the PowerPoint I have, which I'm gonna try to get through this in the next like 15 or 20 minutes, and then I'll explain your lab activities and let you guys figure out what they will include. So let me pull up this. Okay. So exercise 2 has to do with the microscope. And can you guys see my PowerPoint? Oh. Have any of you guys used a microscope before?
15-some years ago.
A long time ago. See, this is why it would be more fun if we were in class and we'd be using a microscope. So we're gonna look at light microscopes a little bit, just in what a microscope looks like, and then we'll be looking at a cell. So if we had a microscope in front of us right now that you'd be looking for, this is what it would look like, and this is what things are labeled on a microscope. And you should know the basic parts of a microscope. The ocular lens is the eyepiece, is what you look at. When you look through the ocular lens on a microscope, you already are looking at something 10 times magnified. So that's what this 10x means. These ocular lenses are already 10 times magnified of what you'll be looking at. And what you're going to be looking at is on the stage down here. So this is the stage, and you'll put a little specimen on a slide, and you'll clip it into these stage clips. The condenser just shines a bright light through the specimen on that slide. And then these are objective lenses that rotate around. And the objective lenses are different lengths that correspond to another magnification value. So you usually have 4 of these objective lenses on a microscope. And they correspond to magnifying what you're looking at 4 times closer, 10 times closer, 40 times closer, and 100 times closer. And usually the shortest objective lens corresponds to the 4 times. And as they get longer in length, they get higher more power. You had to take this down.
That's times the 10%, right, of your initial
Right.
eyepiece.
So
Okay.
then to get the total of what you're looking at something, you multiply 4 times the 10 that you're initially looking at through the ocular lens. So if you're at the 4x powered, you're looking at something 40 times closer. The 10x, you're looking at something 100 times magnified. The 40x objective lens, and again, they just get longer in length as you rotate them around, would be looking at something 400 times magnified. And if you can get to the 100x and actually focus something in a microscope, you'd be looking at something 1,000 times magnified.
So that would be kind of like how the notes would be written down, the research of the specific, cell or whatever we're looking at.
Yeah, so exactly. So, and I don't know if they would have any pictures of it, but normally if you look at a picture in your textbook of a cell or a tissue, it'll tell you what it is. Oh, this is tissue from the small intestine, but it'll also tell you the total magnifying power that you're looking at it. So it'll have like 400x next to that picture, and oh, they must be looking at it through the eyepiece and then at the 40x. Good question. What else? Those are kind of the big ones. The arm is how you would hold it. There's a base. Here's the light source. So the light shines up through here. The iris diaphragm control, this just controls how the light goes through. The light basically goes through it. So you can control how much light goes up into your specimen, and the condenser can kind of condense that light onto the specimen itself. So basically you have a big light source and everything's trying to like get itself into right up through the specimen, which would be on that clip or the slide, excuse me. The light intensity knob controls the brightness, the main on and off switch, and then your coarse and fine adjustment. Your coarse adjustment knob is what you would use to try to find your specimen on your slide if you're looking at it with scanning power, and we call the 4x objective lens scanning power. And then you would use the fine adjustment knob. Once you can locate something with a coarse adjustment knob, that just raises the stage faster or lower to try to get it into focus. The fine adjustment knob would just fine tune it and help you focus it even better when you're using higher powered objective lenses. This oil immersion, usually you would immerse your specimen in oil to help see it better under such a high magnifying power. That's what oil immersion means. That's probably the most boring explanation I've done of microscopes without showing one. Yeah.
Is that data needs to be included on the research paper?
Yeah, if you were like taking like notes in a lab or you were working in a lab, you'd say what objective lens you were looking at.
But not so much with the last part you explained.
Yeah, you could say you immersed it in oil to help future people who might be looking at that slide. Say, oh, you might want to put oil on it to help you see it better.
Yeah, I have a buddy that does rats, like they do drug testing and stuff like that. And when he's talking about it, I'm like, English, dude, English.
Yeah, that's so cool. So he works in a lab, or?
Yes, he works in a lab.
Cool.
He focused mainly on the brain, but he does the whole rat. Like he cuts everything on the rat. They literally put it there and then they cut it like little tiny slices. It's
yeah,
crazy.
That's so cool. So that's why you're taking this class. You want to do that too someday, right?
Well, I'm doing the X-ray tech program, but it just
good for you. Yeah.
It's interesting when he talks about it. I'm like, huh.
Good for you. That's really so. Yeah, I'd be curious to hear. Yeah, I wonder what he does with drugs, but that's really interesting.
Well, he does the test that has each kind of drug and how addictive it becomes and things like that.
Oh, very interesting. Yeah.
And the functionalities of the brain and, and neuroplasticity for the animals and stuff like that.
Very interesting, yeah. And they use rats and mice because they can they're easily they produce themselves really quickly. I used mice in my research program, and so I still hate mice and rats. So these, if any of that didn't make sense that I was explaining, here's the component and the function of it too. Here it gives you an explanation of those 4 objective lenses and then their purpose of how you would kind of use it. And basically, as you work your way up, you're using the 100x for very small specimens. Total magnification, we talked about, is always taking the ocular lens and multiplying it by what objective power you're looking at. And then what adjustment knob you would use, whether it's the coarse one, which is the larger one, or the fine one, which is the smaller one. That just raises the stage up more slowly to help you focus in on what you're seeing. So you should understand how to calculate out total magnification. And basically you just do it by taking your objective lens power and multiplying it by 10, which is what the ocular lens piece is you're looking at. Okay. So if you were looking through something, you'd always start with the lowest objective to try to actually focus in, and then you'd zoom in on it using higher powered objective lenses. So this would be using the 4x, the 10x, and the 40x objective lens. And again, they're multiplied by 10 to give you the total magnification. And this is kind of what you would see. I think they're looking at, I think this is a look at some kidney slides. We'll talk about how you can find that out, but these are kidney tubules. So you're looking at some kidney tissue here. Okay, so then we get into a cell, and I saw a chat. Okay, so I think the rest of this just goes through the basic parts of the cell, which we'll also go into more in lecture. So I'm assuming in your lab exercise in that virtual lab, you'll have practice labeling the parts of the cell. And we'll go more over the parts of the cell in lecture. But here's a look at the cell. Here's the nucleus of the cell, which is the brain. You have mitochondria, which are what create the energy, ATP. Centrioles are these 2 guys. They help in cell division and pulling the chromosomes apart. Secretory vesicles will eventually be excreted. Cytoplasm is the word for anything within the cell membrane. And the cell membrane is just the wall that makes up the cell. The rough endoplasmic reticulum is flattened, kind of tubular sacs, and it's studded with ribosomes. That's why it's called rough. And smooth endoplasmic reticulum does not have ribosomes, so we call it smooth. The ribosomes help, you could also see ribosomes free floating, and ribosomes just help with protein synthesis. Smooth endoplasmic reticulum helps with detoxifying drugs and alcohol, so you'll find a lot of smooth ER in your liver. The Golgi apparatus takes all of the proteins that have been formed and it packages them, redistributes them, and sends them either out of the cell where they're needed or in the cell. So I call the Golgi apparatus, it's like an Amazon distribution center. Amazon's taking over the world. They have airplanes now. Did you know that? We should have all invested in Amazon. Like, good grief, we should have invested in them before COVID, like a year ago. They're buying up shopping malls and turning shopping malls into warehouses. But think of the Golgi like your Amazon distribution center. So it takes everything that the ribosomes have made. So it takes all the proteins in, and then the Golgi sends it out and packages it. What else? The nucleolus is the center of the nucleus. Peroxisomes also help with detoxifying, specifically free radicals of oxygen. And lysosomes help with cell digestion and cell waste. And I think just the next slide takes you through all those structures I listed and their functions that I'll let you guys review on your own. So those are the 2 exercises. And I'm gonna go ahead and just stop the recording. Yeah, I'm gonna stop the recording.